DNA is constantly being replicated and experiences its fair share of error or mutation. While this has yet to cause any superhuman powers, it is a driving force for genetic variation. Point mutations are mutations that result in the substitution of one base for another. Types of point mutations are missense, nonsense, silent, and the readthrough. These mutations can also be classified as transition or transversion. Transition is where a purine is switched with another purine, or a pyrimidine is switched for another pyrimidine. Transversion is where a purine is switched with a pyrimidine, or a pyrimidine is switched with a purine. Here's our coding strand of DNA, which gets transcribed into messenger RNA. Then the mRNA is translated into a polypeptide chain of amino acids. What happens if this nucleotide changes from an adenine to a guanine? It causes a missense mutation to happen. A missense changes the codon to code for a different amino acid, in this case asparagine to serine. This example is a transition mutation because the adenine, a purine, is changed to a guanine, another purine. What if this guanine is changed to an adenine? The second codon will now code for a stop instead of a tryptophan. This is known as a nonsense mutation. This example is another transition mutation. A nucleotide is changed, but the codon product is not. It is known as a silent mutation. In this example, a uracil is changed to an adenine, but the codon still makes an alanine. It is also an example of a transversion, since the uracil is a pyrimidine that is swiped out for the purine adenine. The last point mutation is the readthrough. This is when the stop codon is altered to code for an amino acid instead of a stop. The ribosome doesn't know it should stop and continues to add to the protein. This example is another transversion, since a uracil has been changed to an adenine. Here is a quick review of the point mutations. Thanks for watching.